Hello everyone and welcome to Chapter 19, Part 3A, Gel Electrophoresis. Today we'll be talking about an application of gel electrophoresis as well, which is genetic fingerprinting or aka DNA profiling. And this is the sort of things you see in criminal investigation type movies or series. And you can imagine, you can just hear them talking, right? When the forensic scientist says, the DNA of the suspect matches the DNA we found at the crime scene, and therefore the suspect must be at the crime scene. Okay, anyways, the method that they are describing, the technique used to come to that conclusion, is really this technique called gel electrophoresis. And the function is really to separate fragments of DNA according to length. Gel electrophoresis can also be used to separate different proteins. So there are two types, one for DNA, one for protein. And for the proteins, it's the same function, separate different proteins according to mass or charge. So, just so we are clear, in your syllabus, we focus mainly on DNA gel electrophoresis, and the protein one, not so much. We only touch on it very, very briefly. The focus is the DNA gel electrophoresis, alright? Now, before we go into how actually to determine whether DNA of the suspect matches the DNA at a crime scene, we need to first know what we need for gel electrophoresis and how it works. Let's do that right now. So what are the components we need for gel electrophoresis? If you realize all the DNA methods ones are in red and all the protein methods for gel electrophoresis is in blue, color coding. Anyways, the first component you need is a gel. So it's kind of like a jelly-like substance. In DNA electrophoresis, we use agarose gel. In protein gel electrophoresis, we use polyacrylamide gel. And different gel would have different impedance, and that's why there are different ones for different substances. But both of these gel has even spaces between gel molecules, so DNA and protein can move within the gel properly. So it's not like it's kind of smooth transition, migration, okay? And both of these gels also have wells, and wells are these holes here in the gel in order to load your sample in. So this gel here is actually like around 1 cm-ish thick, and then it has holes in it to put the DNA and protein in it. Those are called wells. Okay, other than that, you need electrodes, specifically a negative electrode and a positive electrode. The negative electrode is called cathode, the positive electrode is called anode. And but for easiness sake, you can just stick to negative and positive, that's fine. And basically an electric field. And why is that? Now, this is the basis of gel electrophoresis. DNA is negatively charged, and we will see this in the slides later as well. The DNA is negatively charged, and that's because it has phosphate groups. And since it's negatively charged, it will be attracted to the positive electrode and therefore migrate from the wells here, repelling against negative, right, and attracted to a positive electrode. If it's bigger, it will move slower. If it's longer, it will move slower. Whereas if it's shorter, it will move faster. So this is the general principle about gel electrophoresis, which we will talk about even more later on. But yes, you need electrodes and electric field for that purpose. Next up is a buffer solution. A buffer solution maintains constant pH, and actually it also conducts electricity. It's not on the slides here, but here we go, extra knowledge for you. It conducts electricity throughout it's actually filled, you can see the level here. It covers the gel as well, the entire tank, so-called. And yeah, so electricity and the electricity field can be moving around this solution. I don't teach physics, so I'm a bit fuzzy on that part of my knowledge. But you know it better than I do, right? Okay, moving on to the next thing we need for gel electrophoresis. Okay, this is where things get a little bit more technical. Don't worry if you don't get this on the first time I say it, because we are going to be using these components later on in our procedure. So just conceptual right now, right? understanding right now. So next thing you need is a loading die. Loading die is a die that you can see with your naked eye. So I'm just going to draw an eye here. You can see with your naked eye, and you can use it to track DNA migration. Usually you mix it with sample. Wrote it out of order. Right, mix it with the sample, and then you can observe the loading die as it moves down. Move towards the positive electrode. And you need this because don't forget, DNA is not visible with the naked eye. DNA cannot see with your naked eye. So you cannot really track whether it's moving or not. And you cannot track if electrophoresis is done or not. So yeah, you mix it with the sample again, and maybe it's here, you cannot see it, but you can definitely tell your loading die has reached the end, and therefore you should stop the gel electrophoresis before the DNA or protein runs off the gel. You don't want it to move towards a positive electrode and then move off the gel into the buffer solution. You want it to stay on the gel in order to analyze it later. Okay, so that's the loading gel. One more thing that we mix together with the sample is actually the staining agent. So loading dye, as I told you, is visible, so you can track it as it goes along. It's just like some dye that moves really quickly. Okay, but staining agent is not visible under normal circumstances anyway, like not with your naked eyes, in natural lights. It's only visible under UV light. An example of this is ethidium bromide, and this is the only example you learned. So remember, ethidium bromide is a staining agent. What does it do? It stains the colors DNA or protein. Okay, this one is staining for protein. This one stains DNA. Sorry, DNA. And what it does is that it binds to double-stranded DNA specifically, and if you cannot see it at first, but if you put it under UV light, you will be able to see the bending patterns as such here. So this visual here is due to this staining agent. Right, I'll repeat that again. You take the sample, you mix it with loading dye and the staining agent, and you put it into the well, which is here. I don't know if you can see. You put it into the well, and you cannot see anything when you are actually doing the gel electrophoresis. You can only see the loading dye. You cannot see the DNA. You cannot see the staining agent. Loading dye, right, is to track the DNA migration. It is also called a tracking dye, if you are curious. And basically, it's visible, and you know when to stop the gel electrophoresis. You know when it's complete using the loading dye. And then you take this gel after you completed gel electrophoresis, and you see it under UV light. And because you mix the staining agent in at the first place, the staining agent binds the colorless DNA, and you can see this bending pattern later on. Nice. Alright, there is one more thing you need here in order to get this kind of results. Here, this little ladder here, okay, is called, well, it's a DNA ladder. It's also called like other names, but let's stick to DNA ladder. It has DNA fragments or substances of known length or mass. So you can estimate lengths or mass samples by comparing them to the ladder. So the DNA ladder already has everything you need in there. Okay, it already has a staining agent and whatnot. You just put it into the well, and you can see here that can I zoom in some more? Yep. You can see here that it nicely spreads out across your gel, and since you know that it comes with instructions, so you know what base pair, so how long each DNA fragment is, so we can look at A and go like, oh, it's the same level as the DNA level at 1000 base pairs, and therefore this fragment here has to be 1000 base pairs. And we can do the same for every single band. So B here, this band seems to be the same level as this band. So this band must be around 800 base pairs, and so on and so forth. So basically, you are comparing your samples and the level it is at with the DNA ladder in order to estimate its length. Okay, so that's the DNA ladder. There's an equivalent for protein ladder, which looks something like this. Obviously, this is not DNA anymore. It's not in base pairs. This is size. This is size in kilodaltons. Moving on. So the next thing we need is gene probes, and it is to locate a specific DNA sequence. Now, what are gene probes? Gene probes are single-stranded DNA. It's made of single-stranded DNA, and it can complementary base pair to the specific DNA sequence you want. Okay, for example, look at this gel electrophoresis right here. Sample A is 1,000 base pair, but how do you know it's that specific DNA sequence that you want? It could be anything that's 1,000 base pairs, right? So what do? You make a gene probe that's complementary to that specific DNA sequence, and then you make it bind. If it binds to A, this means, well, you got what you wanted. But if it does not bind with A, then it is not the specific DNA sequence you want. So, what is the application of that? Where is the rest of the details? Well, it is in the procedure, which is coming soon. So, components so far. Let's go back one slide. Okay, so far we need our gel, we need our electrodes, we need an electric field, we need buffer solution. This is like the minimum, right? This is the basic setup. And then you need the loading dye, the staining agent, mixed with the sample, put into the well. You also need a DNA ladder. You put it in the first well usually, so first lane here, in order to estimate the length of the samples. And you need something called a gene probe, which you don't really know what it does yet. Don't worry about it. Now let's just look at the procedure and find out what happens. Okay, brief. Components, right. How do we put them together in a series, a sequential manner? Let's go. Number one, first of all, if you are dealing with a crime scene, for example, or whatever you want to do, you have to obtain a sufficient quantity of DNA. It is possible to take a very small sample of DNA, amplify it using PCR or gene cloning, which is the previous two techniques, and then only extract the DNA and use it in gel electrophoresis. Okay, so amplify it first, then use it here. Number two, you're going to cut the DNA into fragments using restriction enzymes, so that, well, it can separate into fragments. You don't want it to be one huge chunk, you want it to be small bits. And this usually results in many different fragments of different lengths. It's not all going to be the same length. Number three, my numbering system is problematic here. DNA samples are mixed with the loading dye and the staining agent, as we talked about just now. Loading dye is again to track the migration, and staining agent you will see under the UV light later. Number four, again, numbers are problematic. I will correct it in the final slides. Gel is covered with the buffer solution, okay, and you put the DNA fragments into the wells at the negative end of the cathode. Okay, you mix it first, you cover the gel with buffer solution, then only you put the DNA fragments with the staining agent and whatnot into negative end. So since you put the staining agent in already, it will appear this amazing blue color. It's the color of the loading dye. Yep. So you do that, and what happens is also at the same time you are loading a DNA ladder in the usually first lane, and it's run through the gel at the same time together with your other samples. So it probably looks like this. You have this as a DNA ladder, then this is sample A, this is sample B, and this is sample C. This is usually the case. Then a direct current is applied through the gel. You just switch on the current, and what happens is that DNA would be attracted towards the positive electrode, aka the anode. You can stick to positive electrode, and separation will happen due to the electric field. DNA is negatively charged again, and gel acts as a molecular sieve because it has this evenly spaced molecule in the gel, right? So shorter fragments will be able to move faster and further per unit time through the gel than the larger fragments. So faster and further, meaning that this band here at the bottom of the gel is the shortest, whereas the band that's closest to the wells here are the longest fragments. Okay, this is due to the gel impedance, and this causes the DNA fragment to separate and arrange in order of size. Okay, that's all very chill, right? You get that. Next, you're supposed to visualize the bands under UV light. Okay, when it's all done, you stop the electrophoresis, obviously, and then you visualize it under UV light, and staining agent that you added in the beginning would cause the DNA to fluoresce. It binds to double-stranded DNA, it glows in the dark, it's beautiful, like the first light you saw, it's like greenish, and each band is millions of pieces of the same size, by the way. So this is not one DNA, this is millions of pieces of the same size in one place. And then to find out which length is it, you have to compare it with the DNA ladder. So as I said just now, see A and this DNA ladder is kind of the same spot. So this is 1000 base pair, this here is 800 base pairs, this here is 200 base pairs, this and this, same level, 300 base pairs, this here and this here, 600 base pairs. This lines up, and this lines up to 100 base pairs. Sometimes the bands kind of lie in the middle of two known fragments, so you kind of like estimate. It's just a rough estimate of the length of fragments, really. Okay, this is what it looks like under UV light. It glows. It looks beautiful, by the way. But we're not done. We got nice patterns already, but sometimes we really need to know what is the specific DNA sequence. So to do this, number 8, we take DNA pieces. Okay, we take this gel electrophoresis bands here. Each of these are DNA bands here. This one is the ladder. So we actually transfer it using a membrane or nylon or whatever absorbent paper, and we transfer it onto something else. This is called Southern blotting, by the way. Not very important, but we transfer it to a membrane, so it's like a piece of paper or absorbent paper with the DNA on it already in the bands, yeah, and we heat it to separate the strands. So you gotta understand that this band here, again, not one DNA, millions of DNA pieces, and they are all still double-stranded DNA as you know it. But after transferring to a piece of paper of some sort or membrane of some sort, you heat to separate the strands, and then you cool it and add the gene probes at the same time. Okay, what are gene probes? Now we have the elaboration here. Gene probes are used to locate a specific DNA or sequence of gene, and they are made of short single-stranded DNA, or in short we call it ssDNA. And usually it has a label, okay, it has something attached, usually a radioactive substance or a fluorescent substance. And basically because it is DNA, we can make it to be complementary base pairing with specific DNA fragments we want. So we want a particular, say we want to identify a particular allele, which a particular DNA sequence, okay? So we can do this, we can make it, and when it binds to the target DNA strand, which is now single-stranded because we heated it just now, it will light up, but not under your naked eyes, under UV light or X-ray film. So this is what happens if you use a radioactive phosphorus isotope to label your gene probe. It will look like black and white like this. If you use a fluorescent probe, it will light up in like a green color fashion. Okay, it could be other colors also, depending on what dye you use, what kind of marker, fluorescent marker you're using here. Attach. You can see here the word marker is used again, and again, marker is the idea that you want something to be visible to you, something to report to you. So it could be a gene, it could be just a fluorescent particle like this. So yeah, now that we've done all that, we're pretty much done with our gel electrophoresis of DNA. And yeah, again, this gene probe technology is not always used, but is used to locate specific DNA sequence if you are looking for it. Alright, and I just want to really recommend this site here to simulate gel electrophoresis. It walks you through the concepts and it kind of gives you experimental methods in order to do gel electrophoresis as such. And it has really cute animation, really cute music. It sounds really retro. And it's more detailed and more accurate, closer to real life than any of my slides are. So yeah, try it out. So we're done with gel electrophoresis of DNA. I hope that's clear. Let's move on to gel electrophoresis of proteins. So again, the focus is DNA. Proteins is the side focus, like side note sort of thing. So here is just one slide to tell you in briefly how this works. So for gel electrophoresis of protein, we also call it SDS page. This is not essential for you to know, but it's to separate protein according to mass. Okay, instead of agarose gel, we use polyacrylamide gel electrophoresis, so that's why this is called page instead of agarose gel. And we usually heat proteins with a chemical called SDS to eliminate charge. There are many other types of gel electrophoresis of proteins, but this is the one that's most commonly used. They eliminate charge, so the proteins doesn't get attracted to the positive electrode because of the charge. It's moving because of the size. And just like DNA electrophoresis, smaller proteins will migrate faster and larger proteins will migrate slower, and therefore in the end you have a bending pattern that has seen. Okay, and don't forget your protein ladder here that you can also compare with the sample in order to estimate length. And this is actually from a scientific paper for rails, and you can see here that they identified that this particular band here is actin, and this particular here is MHC, which is a histone complex actually. But yeah, cool, right? It's pretty cool. I think it's pretty elegant. Whoever who came up with gel electrophoresis is a genius. Very elegant method indeed. Okay, now that we have done gel electrophoresis for DNA and protein briefly, let's talk about applications of DNA gel electrophoresis. Again, our focus is on DNA gel electrophoresis, not protein. Ignore protein, that's fine. Now, what are the applications of DNA gel electrophoresis? Number one, you can use it to distinguish between alleles of a gene, so you can break the DNA up. You can gel electrophoresis it, so separating the different fragments, finding the fragment you want using the gene probe, which binds to a specific sequence of DNA. Because it binds to a specific sequence of DNA, you can find the allele. Number two, you can do genetic fingerprinting and DNA profiling, and this is commonly used in criminal investigations, as we said at the beginning, and also paternity testing, so finding out who the father is. Now you might be thinking, why not maternity testing? Okay, maternity testing also can, but the thing is, usually the mother would know if the kid is hers, you know, because the mother gave birth to the kid. It's the father that may be unknown. So paternity testing is way more common than maternity testing. Why would you have to do maternity testing? Anyways, so that's why it's here. Next two applications I'm going to be covering next video, and it's not really just the application of gel electrophoresis. Genetic screening and genetic therapy, gene therapy, requires knowledge from all three techniques you've just learned. So PCR, recombinant DNA technology, as well as gel electrophoresis. So I'm looking forward to teach it next video. But for now, let's talk about genetic fingerprinting and DNA profiling and its procedure. So let's go. Okay, so genetic fingerprinting and DNA profiling. First of all, it is an application of the gel electrophoresis technique, which means we are using principles of gel electrophoresis and using it in a certain way to get what we want. Okay, so in general, is this very simple one. I'm going to talk about more complicated version soon, but it's extract the sample, extract the DNA, make more of the DNA, make sufficient ones, cut the DNA, gel electrophoresis, and actually use gene probes in order to find those specific DNA sequences you want. Okay, let's start with the specific sort of methods. Number one, extract DNA from tissue samples. This could be blood, it could be hair roots. So if you are in a crime scene and you can see those in movies, right, they look for the hairs. Actually, the hair doesn't contain any DNA in itself. The thing that contains DNA is the hair root cells. So the hair follicle cells, those who have the DNA can take that out, take the DNA from there. This could be semen or saliva as well. Saliva itself, again, doesn't have DNA in itself, right? It's usually just water and some enzymes. But in your saliva, there's also some cells, some cheek cells that go in there. So maybe you can extract a tiny, tiny bit of DNA. Right. Now with this DNA, we would identify up to 13 VNTR regions. So in real life, this is already identified, by the way, but for your sake, for your background knowledge, VNTR regions are variable number of tandem repeats. They are short lengths of repetitive non-coding sections of DNA, so they don't code for anything. It's just in our DNA code, and somehow each individuals have 2 alleles for each of these VNTR regions, and the number of repeats differ between individuals. So you can see here, this guy has 2. This is one VNTR region, one type of VNTR region, 2 alleles, one 7 repeats, the other one 6 repeats. This fellow has 4 repeats here, 9 repeats there. So you can see here, two different 2 alleles for each VNTR. Number of repeats differ between those 2 alleles, as well as between different individuals. And this is just one VNTR region. Think of 13 different regions. If there are 13 different regions, it's almost impossible that individuals would have the same one. Okay, so unless you are like identical twins, and that means you are genetically identical, then you will have exactly the same VNTR repeats on each allele for all 13 regions. If not, it differs between individuals. This is why this is called genetic fingerprinting. It's like a fingerprint. A fingerprint is unique. So is the number of repeats in your VNTR regions. Okay, so in real life, these regions we already know, and we skip to the third step quite quickly. So you extract the DNA just now, and then you increase the quantity of DNA by PCR, okay, of the human being, okay, because usually when you get it from a crime scene or even a blood sample, it's kind of like a minimal amount. PCR, make many million copies of it, fragment the DNA using restriction enzyme, cut it all up, okay, not all up, into different fragments, and then we're gonna do gel electrophoresis. And what this does is that it separates the fragments that you just made into different sizes, and after gel electrophoresis, you transfer it to a membrane, you heat it, you add the gene probe, so the whole hoo-ha, right? And then you visualize it under UV light and X-ray film to see a bending pattern. And the bending pattern you see will only be of those VNTRs. Why? Because you are using gene probes which are complementary to the VNTR regions, and therefore that specific sequence of DNA, specific sequence, is the only thing that lights up. The rest doesn't appear. Okay, and you'll get something like this. Right, you have individual 1 here and individual 2 here, and locus A is basically the first VNTR region we're going to look at, and then other than that we have locus B, which is the second VNTR region we'll be looking at. Now for individual 1, you can see for locus A, one allele has 5 repeats, and the second allele has 2 repeats, and therefore you can see the name here, allele A as in locus A, 5 repeats, allele A again for locus A, 2 for 2 repeats. Now let's look at individual number 2. Let's look at locus A again. GC repeat, you can see it says here, this allele has 3 repeats, and therefore the allele name here, A for locus A, 3 for 3 repeats, and allele A4 here has 4 repeats of GC. And you can see the pattern for locus A here in this region right here. So you can have locus for individual 1, there's allele A2 and A5, so 5 repeats is obviously longer, and therefore it migrates slower, and therefore it's nearer to the well, so the well's on this side. And A2 is much shorter, so it migrates very fast, so it's much further. So there are 2 bands here because this individual is heterozygous at this locus. Now, individual number 2 is also heterozygous at this locus, but, okay, the number of repeats is different compared to individual 1. You can see here, allele A3 is migrating faster than A4, but not as fast as A2, right? And A4 is migrating slower than A3, but not as slow as A5. So it kind of hangs in the middle here, showing that individual 2 and individual 1 has a different length of VNTR at both at the same locus. So different number of repeats at the same locus. Now let's look at the second locus, which is locus B. This is the second VNTR region, and now we're looking at a AGCT repeat. And you can see in individual 1, this individual is homozygous at this particular locus because the allele at locus B, both alleles have 2 repeats, whereas for individual 2, locus B is heterozygous. Individual 2 is heterozygous at locus B. So one allele has 3 repeats and the other has 2 repeats. And this is also reflected again in the gel electrophoresis banding pattern that we see here. Since it's homozygous, it's the same length, and therefore the band here will be the same band. So this band here actually is both of these alleles together. Okay, only one band is shown here. Whereas for locus B, B3 and B2 is shown, okay, and it's heterozygous at different length. B3 is longer, therefore migrates slower. B2 migrates faster, so it's nearer to the bottom here. So yeah, that's how you analyze gel electrophoresis when it comes to VNTR regions. Now, just a reminder here, each band is millions of DNA. Okay, even though I said, hey, this guy here, this is heterozygous, so B2 is for allele B2, I don't mean there's only one copy of allele B2 at this point. There are millions of copies of allele B2 at this band itself. It's very, very small. A lot of it is needed to show us a band. In fact, in gel electrophoresis, if there's very little DNA at that particular band, it will be a very thin band. Whereas if there is a lot of DNA at that band, for example, there's 2 alleles at this band, it will show a thicker band. Okay, so quantity of DNA at that particular band would depend on the thickness of the band as well. So yeah, moving on. Let's talk about applications. As we said just now, for genetic fingerprinting or DNA profiling, this is used to find relatedness with other individuals, so how closely related one is from the other. So this is true in the case of paternity testing. In paternity testing, the mother is usually known. That's why maternity testing is very, very rare. And a child usually inherits 1 allele from the mom and 1 allele from the dad. So when it comes to paternity testing, the child would inherit also usually 1 band from the mom and 1 band from the dad. Okay, don't worry if it's not in equal amounts, if you know what I mean. Okay, let's just look at this example first. I have to zoom out in order to write. I think you can still see anyway. Okay, so this child, the mom doesn't, this child, let's look at this child here. It only has, this band here is not present in the mom, so it must be inherited from the dad. This band here is present in the mom and in the child, not the dad, so must be from the mom. From the mom. This one must be from the dad. This one must be from the mom, because only mom has it. This one must be from the dad. This one must be from the mom. Okay, this one's so off. This one must be from the dad. You can see here 4 bands are from the dad, same as the dad, and 5 bands are same from the mom. But don't worry about like how it's not equal exactly, because don't forget that for some alleles, for some locus, they can be homozygous. Okay, so it might, one band might tell you information about 2 different alleles in one band. So it's okay if it's not exactly the same number of bands from the mom and from the dad. Of course, another application of this is criminal identification in forensics, and this is kind of straightforward. Look at the crime scene DNA, take DNA from a few suspects. If the DNA matches the crime scene almost exactly, most likely suspect 2 here is the fella at the crime scene. I just want to say that just because suspect 2 is present at the crime scene doesn't mean suspect 2 did it. It just proves that suspect 2 was there. That's all. Okay, next example is this, right? This is our last example. This is probably a common question as well, where the mother and child DNA is given, and then there's that 1, that 2, that 3. So multiple DNA samples that are taken here from multiple males, and us evaluating which is the dad. This is kind of like a Mamma Mia situation, if you get what I mean. Anyways, let's look at this child's DNA. You can see here that this allele here might be inherited from the mom. This one, don't know. This here might be inherited from the mom. This here might be inherited from mom. Okay, the other 3 bands we don't know. So let's look for this band. This band is only present in that tree. Okay, so most likely that tree already. And this band here is only present in that tree as well, whereas this band here is present in that 2 and that 3. Now, the child cannot just have DNA from nowhere, and cannot just have DNA that's not from the mom and not from the dad. There's no third source of DNA here. There's only 2 sources of DNA for each human being, right? So that 2 doesn't have all 3, and therefore it's not the dad. That 3 has all 3 of these bands that is the same as the child, therefore it's most likely to be the dad here. Okay, so I would leave you to practice more of these concepts and practice how to analyze banding patterns in your quiz. So make sure to try out the quiz right after this. And also in past years, things can get a little bit more complicated than just looking at banding patterns. And of course, I'll be working through some of the examples with you as well in chapters, in worksheets to come. Just one more thing before we end. I don't know if you realize, but there is a paper 5 marking on many of these slides, and that is because Electrophoresis has come out in paper 5 before. Not that it is not like paper 4 cannot come out, but paper 5 has came out of it before, so it might be something you want to study beforehand, before paper 5. Yep. So yeah, that's it for me today. I'll see you next video. Bye bye.